Hi guys, good morning, and today we will finish off amino acids. Remember, last time we met, we talked about what are amino acids, right? And how amino acids can form this thing called Zwitter ions. You can see from here, Zwitter ions, okay? Zwitter ions, right? Remember, amino acids has got two parts. Amino, NH2, and acid, which is your carboxylic acid, COOH. And this in pH of 7, which is neutral water, they will undergo ionization, or they'll form a, here, H here, the positive here will be, well, just rub it off now. No more this H here. Don't know why it's not coming up. Never mind. Okay, coming up. Well, all right, and of course, it will be a negative sign here. So you remember that this is positive, negative. It makes the whole thing neutral. Of course, depend whether the whole thing is neutral or not depends on how many of these groups are there, the NH2 groups and COOH group. If the 2:1 or whatever, if it's 2 NH2 to 1 COOH, that means it will be charged positive. If it's 2 COOH to 1 NH2, it will be charged negative. If they are equal, it will be charged neutral 0 at pH 7, and that pH 7 is called the isoelectric point. Okay, isoelectric point is actually the pH. All right, so I've gone through the whole process with you already. So now we talk about, well, describe part B here, describe the formation of peptide bonds between amino acids and to give it di and tripeptides. Now this is the beginning part to go to your proteins, which we will do later. Okay, but so this is the formation of two join together, two amino acids join together to form a dipeptide. Di means two, tri means three of them. Okay, and you can see how they are joined. Of course, they do it by this thing called condensation reaction. Okay, they ask you on condensation reaction. Okay, all right, which means the water will come out. And then lastly, they ask you for, well, if you have got this dipeptide and tripeptides, can you separate them? Can you bring them up back to your amino acids? If you separate them and go back to your amino acids, can you identify them? Can you separate them and identify them? Yes, we have this process called the electrophoresis. And if you have pH, okay, so we're going to look the whole process here. Hopefully we have time enough to finish off nicely. I've got a lot of questions here ready to discuss with you. Now, first, of course, we go to the formation of peptide bonds. Peptide bonds, or something is called amide linkages, okay, or peptide linkages. This one here, they use peptide because it's on proteins, okay, right? They use amide linkages is for nylon. You can see that, but it's the same thing, okay? So peptide bonds is this one here, like that. Okay, and you see that one we done it before. Okay, now this is called peptide bond or peptide linkage, right? All right, found in, of course, your amines or amides. Okay, so now let's look at some of this question, and then I'll discuss the theory from there. Okay, 214. All right, 214. Okay, now here's a simple question, right? You can see the structures of 4 amino acids may be represented by A, B, C, and D. Okay, all these structures here, right? You can see here, this one here. Now, of course, this is here. The first A is 1 amino group to 1 carboxylic group. Okay, now this is alkyl group. So this is a neutral, okay? Under pH 7, this will be neutral. Charge, you'll put the total charge here. Total charge here. It's equal to 0 here. Okay, now for this one, for B, you have got 2 acid groups and 1 down here. So it's like 1:2, right? So at pH 7, this will be charged positive, this will be charged negative, negative. The whole thing here will be charged -1. Okay? Now this is 1:1, yes. Okay, but this is a, well, it's polar in nature, delta negative, delta positive. But still, okay, it's negative or positive. There's still 0 here. We won't go into the, the zeta ion here, blah, blah, blah. This is just to give you the idea. NH2, 2 of them here, right? So if it is neutral 7, we will say that it's positive here, and it's negative here. So it's charged +1. I told you that all this is given in the old syllabus is given, but in the new syllabus, you can see that you are given in a data booklet. Now, this is a data booklet in part 10. Look at this part 10. Okay. So here, 10 names and structures and abbreviations for some amino acid or amino acid. Now look at here. Okay, of course you can see here. This is the general formula. Okay, I need to add notes. Put down the black color. Okay, this is of course the general formula. Okay, remember I told you what I gave you was C. Okay, here NH2. Here is a COOH, and it's a H here. There must be a H here, and there must be an R group here. Okay, it's an R group here. So the R group can change, right? So this is R groups here. R side chain. So all these are given here. Okay, so the rest of this group here remains unchanged. Start to try and tell you. And of course, this is given the adenine, aspartic acid, cysteine, glutamic acid, glycine, lysine, phenylalanine, and serine, and triazine. Now, of course, here, well, let's put another color, blue. Now, of course, here you can see that this is where there will be 2 carboxylic acid group now. Okay, here 1 and of course here 1. Okay, here you can see that there are 2 amino groups. Here 1 and here 1. Okay, so they give you the variety here for a good reason. And they put here phenol, look at here, this phenol. Phenol is also acidic, So in pH 7, it also gives negative here. After you can see how they come out. Okay, so we vary of this here, right? So this whole thing is given to you in your data booklet. Let's go back to our original. So with these 4 structures of elements given to you, they ask you, A, which functional group, okay, which functional group are involved, oops, wrong. Which functional groups are involved when a peptide chain is formed? Okay, of course, now I'm going to draw anyways on here just to show you, draw a diagram to show A and C. Forming a dipeptide A and C. Now you don't have to worry, you can put C first or A first, it doesn't really matter. Since they say A first, so you draw the stupid A line. Alright, I'm going to draw it down here. Fast. Mouse is spoiled, so I'm going to use it. Okay. Let's try a bit slower. Okay, here. A, of course, will be, I can just draw it like that. See here. I draw A first, so NH2. Better to draw everything out. This is blah, 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 R. Okay, I put draw R here. And there's H here, and of course, a C double bond O, and draw it out. It's an A. Okay, now they want A and what? They want A and B. Okay, now B, you can see that there are 2 carboxylic acid groups there. So A and C, sorry, A and C. So face to face this amino group with this one here. So this is your alcohol here, okay? So now let's draw the alcohol here. Okay, maybe I draw the whole thing. Okay, so this is actually CH2 and C and CH3 and this CH3 and this H. Okay, so now I will draw CN and here I will draw it out like that. All right, C and H here, I will draw this thing here, CH2OH here. I can do like that. Okay, right? Now box this. Remember, water comes out. You can box it, it doesn't matter. Right? To give you, of course, if you want to put on here, right? To give you this one here, and H. Now you can draw these two here, doesn't matter now because you already know what is going to be happening. CH2 and C and CH3 and H here and CH3 here. Now this is the peptide linkage. C mono to N H here, C H here, and to CH2 OH here, and your COOH here, put like that, plus water. Now this is called a condensation reaction. Okay, condensation reaction. They ask you one. Okay. Now this, you can box right here, is called the peptide linkage. Okay, so this is called, put up here, peptide linkage. Okay, quite clear, isn't it? Right? So you can see here, right? They say, okay, which functional groups are involved in the peptide linkage chain? Of course, what functional group is? These are M functional groups here. Amino group, or amide, or amine, sorry, amino group, and your carboxylic acid group. Okay, draw the structure from, okay, now let's go to the marking scheme. I think it's 10. Okay, which is this one here. Okay, right, so carboxylic acid group, you see here, carboxylic acid group, okay, so, all right, I must find all this thing. Sometimes I wish I could, telepathy, man, I can just tell, hey, do here, carboxylic acid group, right, and amino, or amine group, either way you can say amino or amine group. Okay, formula acceptable, right? 1 mark. And so this one here, see now, I draw here and this one here. You can draw reverse also, it doesn't really matter. Okay, you can put NH, this one here, and then this one will form the bond with this one here. It doesn't matter. Okay, so now let's go back to here and see other examples. This is a dipeptide, because the syllabus say, how about tripeptide? We get. I got one question here for tripeptide. Okay, 215. Okay, the next page. Okay. All right. Now here's spider silk. All right. Well, I think I moved this away from it. Okay, spider silk is a natural structural protein which belongs to the same group as collagen and keratin. It contains a higher proportion of amino acids of glycine and alanine. So they give you here, right? Glycine, famous as CH2. Alanine, okay, so it's a 1:1 ratio, isn't it? 1:1 ratio. Also here, 1:1 ratio of amino group and your carboxyl group. So it's not 2:1. So draw the structure of a tripeptide. Now they want this one first, glycine, followed by alanine, followed by glycine. So the arrangement is very important, the order. Because in all proteins there's an order. The protein from your eye, your hair, all different one, no. Out of the 20, they were like glycine, alanine, trypsin, glycine, trypsin. So the arrangement of the type is all different. So the protein comes from your, from your muscles, from your hair, all different. but they all come from the basic, like it's all A, B, C's, and you form different words as well. And of course, all controlled by who? From your genes, your DNA. We used to do, DNA, actually how the genetic code fix what proteins to mix. So if you got a lump of something going wrong, that means the protein is all mutated, so you get growth of different things in your body. It's like a cancer. Okay, you can read up yourself those things, but we are interested in basically dipeptide and tripeptide, and how to draw the displayed structure of the peptide. That means the peptide linkage must be displayed. Okay, so here 3, so glycine here. So what do we do? I'm going to move it up here. Okay, so easier for you to see. Right, the top part here. I would, okay, easier. Okay, we do, see, no, come on. Okay, that's glycine first, so you put down here, all right? It's good, as I said, if you were to draw the displayed formula. Some of you are not so clever in this structural formulas, so draw like that, you get a mark. But you're clever, is it? You know your stuff, is it? So why do you let them penalize you for nothing? Okay, so here, I draw the second one, which is your alanine. Okay, now some must face with the amino group. So H here, and the H here. Draw face to face so that they can actually box it as a, right? Alanine CH here, CH3 here, and of course, draw the structure out. There's a mosquito here. I'm gonna kill it. Okay, plus now glycine, which is your draw H and H. Draw the whole thing out. Just about 1 second or 2 seconds, you're faster. Okay, H and H here. And okay, remember, don't draw the ionized stuff. And then box. Water comes out. Okay, 3 tripeptide will give you 2 linkages and 2 water come out. Okay, right? Is it draw a structure? Is it? This one is not enough. They want the structure here. So now here, of course, you can, so once you draw that, you can put this one out, CH2 now. Okay, but here you must draw the display structure now. Because you want displayed. So this must be displayed. And H. By the way, this thing here is a flat molecule. No, hold on, I thought I better tell you first. Okay, CH3 here. And H here. And C double bond O. This must be drawn like that. Because they displayed. Okay, C, can put COOH here. Is it correct? Sorry. This is your C glycine, is it? Yeah. H C O H. Okay. So now let's go to marking scheme first. Okay. So what year is it? This is 06. Okay, here we are. 06. Okay, see now. All right. You see this? Let me mark this up there first. So, okay. Right. So now remember, glycine. Now see now. You do here. 2. So for tripeptide, that means 3 amino acids combine or condenses, and it forms one tripeptide and 2 peptide linkages. Very important, they ask you to calculate one linkages, and 2 water given up. Later I'll show you. They will ask you when you do your proteins, they ask you to calculate the MR of the whole thing, right? They give you the individual substance, the monomers, and then they give you one crazy thing here and ask you, well, they all found one big protein link polymer. What is the MR? Okay, so you know, and how many peptide linkages are there? So the number of amino acids -1, you get the peptide linkage. Number of amino acids -1, you get the number of water coming out. Okay, so the display. Okay, 1 mark. Peptide linkage showing up. Now remember, this linkage here is a flat molecule because why? Carbon here is actually sp2. Wait not? Yeah, no, this is sp2. sp2 hybridization. So remember, this is flat molecule. Okay, flat trigon. The angle is what? 1, 2, 0. That's all here is 1, 2, 0. Here's 0, 1, 2, 0. So to end here. Right? And here. Now, of course, N here, this N, okay, right, is sp3. So remember sp3, one of them go here, one down here, okay, and a lone pair here. It's not, okay. So now you see, at least, okay, here, H here, one of the H here and this one here are same plate. This will be out because two are the same one, okay, and this one here. So this is a flat molecule. See, flat, right? So remember this whole thing here is flat, okay. Plus this one here. So you ask you how many items here? 1, 2, 3, 4, 5 of them are in the same plate. So it's off already, okay, right? But this part here is a flat molecule, okay, flat. Okay, but I won't go further. Go back to here. Okay, now of course they ask you what type of reactions are there? Condensation, right? Condensation. Okay, so you can see here they ask you what kind of reaction is it. Okay, what type of reaction? What type of reaction takes place during the formation of the peptide condensation? 4 marks, simple, easy to get. Okay, I won't go to others first time. Now I want to go to now. See, now you know how to what does it mean by dipeptide or tripeptide and a polypeptide. Of course, a polypeptide means a protein. Yes, so in a peptide, linkage is the thing that joins the two amino acids together. Di means 2, tri means 3, poly means many. Now, even though they are formed in our bodies to provide you with proteins, but sometimes you want to identify, like now you know the coronavirus, blah, 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 and they want to find the protein inside there. So they have to now break down the protein to smaller pieces, right? To see what is the composition. To break down a protein, what is it called? Well, to form protein is called condensation. To break down protein is called hydrolysis, because a amide linkage can undergo hydrolysis. Remember that? We did before, is it? Now let's go back to. I'm going to show you the electrophoresis first. 221 here. Okay, I explain a little further. Now this is the whole all my notes on electrophoresis. Now before that, of course, you can see that. Now let's go back to well, what 221 first. What they want and 221. Okay. Sorry. Okay, 132 lah. This problem. Okay. Minus 132. Is it? Yeah, in the beginning here. Now you see here, They say describe simply the process of electrophoresis. Okay, first of all, is what is electrophoresis, and secondly, how does it work, process. And then they ask you, well, how does pH effect of pH, okay, have on the process of electrophoresis? Remember, why is this here? Okay. Now, remember, you make proteins by condensation, all right? You break out proteins by hydrolysis. Now, I'm going to go back to, okay, remember, keep in mind the word electrophoresis. So this is actually the DNA fingerprinting stuff, which the biology will do. We used to do it, we still do it now, but not as detailed as last time. So you're lucky. Okay, 3, 2, 1, let's go there. Okay, so I'll explain to you briefly. Okay, let's see here. Okay, up here. You see, when you form your proteins, let's do it quickly. Okay, NH2, CH, H here, COOH here, plus another one. Okay, let's say OH here, O, C, and C, and H here, and CH3 here. Okay, and COO, and H2 here. Okay, so let's say, okay, now to form this tripeptide, of course, is NH2 here. Okay, CH here. Okay, and of course, this is your peptide linkage. Okay, which is the key thing. I just draw two different, aniline and glycine, okay? COOH. Now, this is called condensation. Condensation, okay, this reaction. If I want to go backwards here, it's called hydrolysis. Remember not? Okay, it means water. You will break this linkage here. That's why proteins can be broken down by trypsin. Is water, and trypsin is just an enzyme. Right? So you got a long chain of protein, blah, 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 blah. How to break it all down? Okay? Your body does it quite well. You use trypsin and water. All right? So they can break down to one individual amino acids, or 3 of them, or 4 of them, or 2 of them together. And your body can absorb all this type. So the more broken down, the better. So you need to chew your food. As you chew your food, they become broken down first in your mouth. Right? So your body can have an easier time to break down further hydrolysis. Remember, in hydrolysis, water participate. Okay, you break here. Right? Which means the H will go back to the original NH2, right, to go back to here. Okay? Here, your original NH2 here. Something is wrong here. I'm sorry. In my cepat, I messed it up. Okay, here of course is this. This should be. I should draw it. See, so make so much mistakes. You hurry, make so much mistakes. Okay, here should be NH2. Right, this is COOH. Okay, so that's right here. Right, so now go backwards. Remember, when you break up here, the OH will go into this carbonyl group to give back your carboxylic acid. Okay, so that's called hydrolysis. Now, if it's acidic, actually, you remember if it's acidic hydrolysis, I'm going to put red now just to make a difference. You will get the H here, and it'll be positive here. Yeah, and it'll be a H here, positive here, right? And if it's alkali, I put another color now. If it's now this is acidic. This is acidic, red. If it's alkali, I put green now, okay, or purple color, purple. If it's alkali, right, and OH. This part don't have. It remains the same, but this part is minus and the Na plus. You're from the salt. So be aware of this. Okay, right? So the type of product you form by acidic hydrolysis and alkaline hydrolysis are different. Okay, you see more later as you deal. They are very specific in the answer. Okay, now let's go down to what is electrophoresis. I will start with a question. Okay, very simply, the question actually asks you to draw. Okay, 230. Many times come already. Last time they used to be your favorite. 230, and ask you to draw this crazy diagram. Okay, right. This is one of the questions. Many questions. One of the key areas of investigation in understanding the structures of polypeptide, that means there are many amino acids joined together. Okay, and the proteins in sequence of amino acids are sequencing. We'll talk about later lah. Proteins are they make up a polypeptide chain. So the keyword, of course, is polypeptide. Okay, topical still with us. One of the methods used to determine the amino acids. That means you break down. Then you break down here. Okay, you undergo what? Hydrolysis. SIS. Normally, they'll ask you how. I remember 666 on the devil, so I mean, this is what they give in the marking scene one. So I took it down. But of course, now they simplify it. Okay, heat for 6 hours. That means reflux long time, 6 hours. Using 6 molar of HCl. This cannot go wrong. That means using aqueous acid. So you reflux, you heat. Okay, you heat for and you use acid. So it's acidic hydrolysis. Okay, so 666 you want to. The other 6 probably is U. And proteins in the sequence of amino acids to make up the product. That means you understand the structures of the amino acids. One of the methods used to determine the amino acids present in the polypeptide, present in the egg, is by electrophoresis. Right? You now break it up. After breaking up hydrolysis, this one to the amino acids. Right? You got mixture here. So what you want to do is the one separate them. Yeah, it's very important to separate and to identify. This is of course is in this thing called identify. What are the amino acids and how many are there? Okay, this is of course in analytical chemistry. Okay, in analytical chemistry. So I better go on faster. So they say sketch and label apparatus used to carry out electrolysis. That means that they say sketch, 4 marks, man. There you look here. 4 marks, okay. Right? So now let's look at the, I think I've got the answer script down here. Okay. Okay. Easier to teach. Now, you're supposed to know this diagram. Of course, a simplified diagram will do. But this is the one they provide in the marking scheme. Sketch and label the apparatus used for electrophoresis. Example, okay, this is for example here. So it's not like, oh, don't do this. Okay, very simple. Now there are 2 trays here, which the electrolyte. Why electrolyte? That means it's liquid. Actually, it's aqueous water and a buffer solution. Okay, see, there's a buffer solution. Electrolyte is a buffer. The pH is important. Buffer you do later in your A2 physical, okay, buffer solution. That means you control the pH, control pH. You want the pH to be either acidic, neutral, or alkaline, up to you. So these 2 parts here are your electrolyte, okay? Electrolyte, which is your buffered solution, okay? That means the pH, No. Now then you put 2 glass slides on top here, like the one you see in microscope one, and you put this the filter paper, okay? Filter paper soaked in the buffer solution, which is electrolyte. Right? Now this is the power supply. Pause it. This is the direct, DC, not AC, okay? But DC. So you put that here. That means this side will be positive. I better use a red color, not clear. This is positive. This is negative here. Okay. So now you put your amino acid mixture here in the middle. Okay. All right. So the point is, in this buffered solution here, will your amino acid be charged neutral, negative or positive, or two negative, two positive, whatever is it? Then they will move accordingly, is it? That's not. So the first thing is this is the apparatus. Now look at the marks for the apparatus. Okay, because the power supply here. Okay, 1 mark. Electrolyte with filter paper. Electrolyte, okay. Filter paper. 1 mark. Buffer. I must say there is a buffer. 1 mark. Acid mixture in the central. 1 mark. See now. 4 marks. Crazy diagram. You don't have to be an artist to draw this diagram. So the mark is given appropriately. Okay, now let's look at the thing here again. Okay, what they ask you. Now, this one is we did before. Okay, now maybe before we do this, I would see this. Yes, question. Now let's look at the question first anyway. Okay, they ask you, electrolysis, different amino acids move in different directions, whether positive or negative, at different rate. Now, two things they ask you for: direction and speed. How fast? It's important. So the direction in which you move, forward or backward, depend, of course, first on the buffers in the pH of the buffer. Okay, I tell what after of buffer. And secondly, what depends on the direction. Whether for this pH, the amino acid is charged positive or negative. Amino acid, the charges is charged positive or negative. Right? It can be charged 0 or whatever. Okay? The factors determine the speed of movement. Of course, how fast is the molecular mass, the MR. Okay? Of course, also it depends on the voltage. The voltage. Okay, a couple of things now. This all will come later. Okay, I can see here the marking scheme. You want to look at it first before I go to the nitty-gritty parts of this here. See, right? All right. The direction is the pH of the buffer. Yeah. The charge of the amino acids. Okay. How fast it is? Size of amino acid MR. The voltage applied, or the magnitude of the charge, +2 or +1. Temperature also will affect all these things. Factors here, common sense factors. Okay. Now let's go back now. I've talked to you about this. I want you to go back and read. Okay. Read the thing. I don't have time to go through all of it. Okay. I want you to go and read. Okay. Electrophoresis 221. 221. I think it's 221. Okay. Now here is all the notes on electrophoresis. Okay. You can see it starts here like that. Okay. Okay. Aim to hydrolyze proteins. That means it's a long molecule, okay, of many, many peptide linkages here, many. If there's 1,000, come from 1,000 monomers or 1,000 amino acids. Okay, so how many peptide linkages are there? 999 peptide linkages. Think for yourself. If you don't know, go knock the head. Linkages. And how much water coming out? How much? Same here. 999 molecules of water comes out. Okay. Comes out. Okay. Figure yourself. You don't know. I think you better go to this form 2 or whatever. Okay. See the idea is to separate and identify. Can be peptides, can be dipeptide, tripeptides, and also individual amino acids. Remember now, this is the monomer. This is the dimer, trimer, whatever. Okay? Technical details I talked about already, and so forth. Okay, can read up yourself. Okay, now I'm going to go straight to the thing here. Okay, I won't go through this later. We can go through later. Okay, so how to hydrolyze, how to break up the protein. Reflux. Now the disease is reflux. Actually, they to be exact, the marking is 6 hours and 6 molar to HCl, or you can use trypsin. Okay, right? So forth and so forth. Here you can see now. Okay, voltage, concentration of agar. Actually, they instead of using the filter paper, you use agarose gel. Okay, because why agarose gel? Gel, which means is semi-solid, means it's liquid. Means the ions can move. Ions are able to be mobile. Ions able to be mobile. They move. Right? You want to move, is it? Is to separate them, is it? Time run, I'll tell you later. Okay, buffer, temperature, and so forth. Get rid of all these things. Actually, the graph is very nice. The graph can be used for paper 5. Okay, 2 types of paper and gel. Don't worry about it. Get rid of yourself. All the things here. Okay, now I want to talk about the results. This is more important. Now here we got now after running the electrophoresis here, I got 3. Let's say I got 3 original amino acids: glycine, all right, aspartic acid, and lysine. Now what are they? Now remember, it's pH 7. I told you pH 7, all the functional groups NH2 will become what? Become NH3 positive. All the carboxylic acid will become what? All remember, become CO minus. Okay, so this is the acceptable fact. A levels, that's it. Okay, of course it's not exactly 7. You can go to university and find out it's a different one. Don't care, right? So aspartic acid, lysine. Now to know what they are, all right. Now let's see here. Aspartic acid. Okay. Okay. Where's aspartic acid? Okay, here we are. Okay, all right, here. So for aspartic acid, it will be, I'm going to draw here, okay? CH2 and then C. See, they also draw reverse then, right? Easier what? To your right, remember? To C here, to a NH2 here. Ayah, tak boleh lah. Kundu. This is very good. Never mind. Never mind. Let's do it again. Sometimes. Okay, let's draw a bigger page. Okay, let's draw the aspartic acid. Remember, take the top one here. Okay, here you are. Actually, I'm going to finish reading on your A2 syllabus already. Last one, of course, is proteins and a bit of carbohydrates, fats, and also polymers. That's it. We did a good job. Or I did a good job. You first never studied. Okay, aspartic acid. Okay. Draw slowly. Okay, that's the one here. CH. And now to the one here. NH2 and H here, and of course here's C. Okay, now this is the neutral form. Now, but in acid, in neutral, I told you all these things here will undergo what? Ionization, isn't it? So, I'm going to mark it off. So here we don't have anymore. Okay, here it's not 2 anymore. Okay, here also no more. Okay, so instead, what is there? Blue. Okay, or it will be a minus sign here. Here will be an NH3. One here, another one here, and a positive sign here. Here will be a negative sign here. So the overall at page 7 is what charge? -1, yes. Okay. Yeah. And lysine here. Yeah. So here you will join to the C and your H here, right? And here. I forgot. My goodness. Okay, your COOH. Okay, right? See here. This is your R group. Okay, so H here and of course your NH2 here. And your carboxylic acid. Yeah. So if it's pH 7, you now must put all the ionized stuff. Oh, sorry. So here, small base. Okay, I'm going to draw here. All right, here I can't. Never mind. Okay, here will be another H positive. Here will be one more H. Here will be negative then. So two positive, one negative. Here will be positive 1. Yes, not. Yes, not. Right. So aspartic acid is negative 1. Lysine is positive 1. Very bad. Okay, now let's go back to here. What's the other one? Alanine, is it? Alanine is just CH3, isn't it? Right? That means if it's alanine, if it says alanine, put CH3 here. Right? This will be negative. It's positive, isn't it? So this is 0. So got three amino acids with different charge: a 0, a negative 1, and a positive 1. So I put them in the middle of the glass slide and I put the voltage on. Where will they move? They will all move in different directions, except this fellow here will not move at all. You know? So that's why they ask you now. So I can separate them. Identify them. See now? See? Right? So here, you can see, right? Glycine, after putting here, glycine, okay, miss HH, not alanine, and glycine here, it didn't move. They ask you why it didn't move. Why? Because it is neutral. Charge is 0. Aspartic acid is negative. It's not. Remember? Negative. Aspartic acid. Okay, don't believe me, look again. Aspartic acid what? What is it? Negative 1, is it? Negative 1. Okay, lysine, positive 1. So you can see, right? Here, aspartic acid, which is negative 1. Overall charge moves to positive and it moves to the negative here. But the distance move also different. Now here is shorter, here is longer. Because why? This is a heavier molecule, lysine. Okay, more branching and all these things will affect the movement. See now? Okay, lysine, right? Lysine is a longer, so many C's here, so many C's here. Okay, so they move slower. So the rate, the direction and the rate. So remember, they asked you the question here. The direction of the movement, direction, right? Depends on the charge of the ion. 2, on the pH of the buffer, right? Of buffer, right? And of course, 2, the rate, right, will depend on the MR, the size, right? The temperature. Okay, all these things will play an important part. So now you see how they ask the questions, okay? Right? You can see here, I explain now. Now here, this one here, triosine. Now triosine, you can see later, I got this one here. All right, now this is a carboxylic acid group, correct? This is your amino group, correct? Okay, now the side chain here has got a phenol group, you know. Remember? It's like this one. Not very good in this. Okay. So OH. Okay, join the CH2 here and join to that common carbon here. Going to be primary and your NH2 here and so. Okay, now see here, I show you triosine. You can see here, triosine. Triosine. I say. Gone up, triosine. This thing keep on moving. I don't know what's wrong with it. Okay, triacid. Any triacid here? Yeah, I got there. See that? You see that? Okay, triacid. Now this OH here, remember, phenol is a bit acidic, isn't it? So you give a negative sign here, no? Okay, so you go back here. So in this case here, it will be like that. So don't have any more negative. And this C, and of course NH3 positive, and H here, and this CO minus. So the O is one negative. Okay, so triacid is one negative, and of course you look at here. All right, so triacid is one, aspartic acid is also negative. So aspartic acid is also one negative. It's one negative at pH 7. pH 7 triacid is 0, right? No charge. This is one negative. So these two fellows are moving in the same direction towards here. But they move at different speed. Because this fellow here, triacid is heavier. Because this kind of bonding here moves slower. Aspartic acid move faster. See? They will ask you why. Because it's heavier. Get up. Right? So you can see this how it works. Okay? All right. Now, I want to draw just one question, then we'll try to finish it, okay? Okay. Pattern protein hydrolysis pH. Okay, let's look at 212. Okay, question 6. All right. Describe how proteins are hydrolyzed so that they can be analyzed by using electrophoresis. How are they hydrolyzed? Okay, of course. What? 6 hours, 66 there. See now? Yeah. Yeah. Reflux for long hours, 6. As I put here, 6 hours plus with 6 molecules here. They say reflux for a long time. Or if you don't use HCl, you use trypsin also can. Okay, so two things are heat and of course your acid. Okay, not alkaline acid, trypsin. Remember, trypsin works very well in pH, low pH in the stomach, so it's acidic. Okay, right. Now here, with the inner diagram, describe the process of electrolysis and separation of peptides and amino acid peptides. Right, it means that can be dipeptide and tripeptide. Okay, of course here you can see they give a very simple diagram. Okay, all right, you can see here. Right, you can see here. Here is also well, here they give 6 marks, man. Of course, they say locating agent and so forth. Of course, let me explain again the locating agent. Now, of course, first of all is the diagram. Kept wet in the buffer, okay, 1 mark here. And then the thing here, this is the, this is filled with agarose, and this is to the what? To the DC batteries, right? Conditions, time, 1 mark. Compare references. I didn't go about it, okay. Locating agent means what? Now here in hydrate, you look, if you done biology, sorry. I must put them in hydrate, right? In hydrate is a locating agent. That means when you spray in hydrate onto, because amino acids are colorless. Amino acids are colorless, so you cannot see where they are. So how do you locate them? So you use a what? Locating agent. And ninhydrin is a very good one. Ninhydrin. When you spray ninhydrin into this space here, they will reveal a purplish spot. Okay, so you can know this. Or you use iodine. It's called a locating agent. Iodine also will release brown color onto the amino acids. Okay, so you get 1 mark for all this stuff. Okay, okay. Let's look at the other part of the question. Okay. Okay, so explain how these peptides are. Okay, now by this method. Actually, what you do is you measure the distance. You need, of course, a reference to measure the distance, and we talk this about later. Okay. Okay, just want to talk about this simple thing to fulfill these conditions here. Now, suggest and explain the reactive movements during electrolysis of pH 7 of the fragment. Now, remember here, simple, isn't it? Now, since they got like glycine here is 1:1 here. So it's 1:1 here. That means that at base 7 is charged 0, isn't it? Yep. Now this is 2 carboxylic acid to 1, this one here. Of course, this will be positive, this will be negative. So the overcharge is what? -1. Right? So this NH2 is positive here. NH3 already hydrolyzed positive, and here's -1. So it's +1. Sorry. Okay. So of course, the relative moment, okay, so just explain relative moment. That means this one will not move. Why? Because it's neutral. Draw the structure out. This will move to the positive pole. Why? Because it's negative here. This will move to the negative pole. Okay, now let's go to the marking schema. Okay. Here again, what's it? 0, 4. Okay, here we are. Now let's look at the marking schema. You aim at this, the others you can look at yourself. Okay, see now. You draw it out. Moves glycine, move because it's positive, right? Because of 2 NH2 groups. Okay, right? Glycine has got 2 NH2 compared to 1 COOH. This one has got 1 NH2 to 1 COOH. This one has got 1 NH2 to 2 COOH. So it's negative, is 0, is positive. Now you also say, move less because it's heavier. Okay, you can calculate out, move faster. So lysine here. So the mark is given appropriately. Clear? Okay. All right. Now there's another question here. Maybe you can try it. Okay. Got 3230. Okay, this is the first one that we talked about. Okay. Electrophoresis. We talked about this already. 233. Let's see. 233. This is the marking scheme. Okay. This is another one. See another one here. The first part, of course, is explanation of how it works. The second part here, amino acid glycine has a formula of blah blah blah, 1:1 ratio. Identify the species formed on the filter paper if glycine moves. If glycine moves to the positive, that means it's charged negative, isn't it? Right? So when will this be charged negative? Okay. High pH, acidic conditions, or what? So see, if remember, if you put acid HCl here, the HCl will go to here, isn't it? Right? So you get what? You get NH, H, and CH2. You must know your chemistry. This will be positive charge, isn't it? And if you put your NaOH here, what happens? This one will, this one here will, but NH2 is a base, remain the same, and this will become, that's not. So, right, the species formed means this one. The following results has determined, obtained from blah blah blah. Okay, that means the size, charges of the acid. Very simple, isn't it? So of course you move to here, A and B are what? See, they ask you for here, you can see. Relative size and charge. Okay, what are the charge first? Of course, these are 2 are negative, isn't it? Negative, isn't it? C is positive. So this is small. Right, move faster now. Yeah. This is medium and this is large, isn't it? Yeah, relative size. So it is. A is small. That means move faster. B is what? Large. C is what? Medium. This is the way you should put that. Okay? Or small, smaller, whatever, up to you. Okay? Okay? So all these ways they can ask you. Right, now one more is quite good one is 227. Okay? Talk about glycine one. You can ask in many ways, but if you know your chemistry, no problem. Okay, here on top here. Okay. Okay, you can see here. Right, try yourself 227. Electrophoresis can be used to separate amino acids which are produced by hydrolysis of a polypeptide. Using glycine as an example, explain why the results of electrophoresis depends on pH. Okay, why? Now, of course, you must explain what is glycine. You don't know, you die lah. Okay, this is the simplest thing. Yeah, glycine, the first member. Memorize it, even though it's given there. So remember here, why depends on pH. So say at pH 7, what is the species? Just put under the ionized state is positive. This CO minus, that means the total charge put on there, net charge are better. Net charge is what? 0, is it? At acidic solution, then acid will take where? Now can start with down here. This is already filled, isn't it? It's on here. It's the base, man. So acidic solution, you get what? You get NH, H, and C, H, and CH, and OH. So the net charge is how much? Positive 1. Yes, in alkali solution, okay? So alkali put OH minus here. So which part of them will act as acid? Of course, you put these two here, this and this not, or you start with also can, right? So which acts because here is not, so it's going to come out. So give N, H, no more the positive sign, and your CH. Work it out yourself for friends. Alkali, right? So this will be O minus now. Yeah. So the net charge is how much? Negative 1. See? Okay. Now let's look at how they actually question it. I think of a thing here. 07. Is it 07 here? Okay, June. This is June. Okay, here we are. Okay. See? NH2 or COH minus, this will be protonated. That means a positive A. They move to negative. This one, this two, or this one lose a proton here, or gain a proton here, move to a positive. Okay, so 1 mark for them, and here acidic will protonate, okay, and so forth. See? Okay, so this is the question now. Here, of course, then you ask the other questions here. Try yourself. All right, now this here. Before, after. Diagram shows the results of electrophoresis in neutral pH 7 at the start. Experiment of spot is in the center here. P, Q, R, and S are placed in the middle of the plate. Following electrophoresis, and it's so here. Obviously, which amino acids exist as mainly as ion in the buffer solution? Of course, here, Q. Why? Because it don't move, isn't it? Because it's not moving. Okay, let's see how the answer is. Okay, and P is charged negative, isn't it? Right? R and S are charged positive, but R is heavier. Okay, assuming so on and so forth. Okay. So explain your answers to all these things here. So it's very simple. Okay, calculated. Okay, now let's see what year is it? 07. Okay, 07. See now. Okay, Q forms ion because it does not move. Okay, it does not move. R is larger, travels slower, and so forth. Okay. Okay. All right. Okay, guys. So we have finished amino acids, everything. The final chapter in your A2 syllabus is, of course, polymerization. That'll be the last one. I'll probably do it depending on my mood. Okay, finish already. All right. So I know holidays that now there won't be any Zoom because none of you got questions to ask. A few of you, that's why I tend to entertain you individually. Okay. So well, let me see how to stop. Okay, stop recording.